Okay, I think everyone's had a chance now to get dialed in. So welcome everyone, and thank you for dialing in to the next episode of the 2023 State of Play series. For those of you on Zoom, you may see a notification about the meeting being recorded. Just click OK or agree, whenever it asks, just to continue and get that little box off your screen. So firstly, an acknowledgement of country. So in the spirit of reconciliation, MND Australia acknowledges the traditional custodians of country throughout Australia and their connections to land, sea and community. We pay our respect to the elders past and present and extend that respect to all Aboriginal and Torres Strait Islander peoples today. And I'd like to acknowledge the Wiradjuri people on whose land I am and pay my respect to elders past, present and emerging, and extend a welcome to any Aboriginal and Torres Strait Islander people on this call today. So as many of you are probably aware, we've had a recent approval in the US of the first genetic therapy for MND. So we thought it would be a good opportunity to have a bit of a deeper dive into the genetics of MND. So we're incredibly fortunate to have two world-leading experts in different areas of genetics on the State of Play tonight. Associate Professor Kelly Williams is a genetics researcher from Macquarie University, and we have Dr. Ashley Crook, who's a senior genetics counsellor, Austin Health in Melbourne, and also holds a research position at Macquarie University, where she recently completed her PhD. As with previous episodes, each presenter will talk for about 20-25 minutes, and then we'll have a combined Q&A session at the end. Please submit your questions through the chat or Q&A function in Zoom or through the comments if you're watching through Facebook. We'll collate all those comments, so don't worry. You can put them through any time during the talk. So if something's right on the tip of your tongue, put it in the chat and we'll make sure we ask that question at the end. You don't have to wait to the end to type in your comments or questions. That's all you need to hear from me. So if you're ready, Kelly, please take it away.
Thanks, Gavin. I'll just share my screen.
Okay, that all looks great.
Okay, all right. Well, thank you very much for allowing me to talk today about the importance of genetics in motor neuron disease. Oh, slides. So I'm the head of Genomics and Bioinformatics within the Macquarie University Motor Neuron Disease Research Centre. And we are a large research centre comprising around 70 research staff, professional staff and students across 8 different research teams. And we have a diverse range of expertise, and we work closely with the Motor Neuron Disease Clinic on site at Macquarie, called Macquarie Neurology, which is the largest motor neuron disease clinic in Australia. And collectively, we're working towards answers for some really important questions in motor neuron disease. And so my research team predominantly focuses on understanding the underlying genetic basis of motor neuron disease. And so I wanted to, before I start, I really wanted just to take a step back to explain some fundamental principles about genetics, particularly DNA, genes, and inheritance. So DNA is our genetic code, and it's the blueprint of life. It's comprised of around 3 billion units of our DNA code, and it's present in every single cell in our body. And so these 3 billion units of DNA, they lie adjacent to each other along what we'd consider, a ribbon of DNA. And these adjacent units of DNA are either we summarize them as an A, a T, a C, or a G. So there are only 4 types of DNA units that comprise our entire 3 billion units of DNA code. And so in order to fit our DNA in every cell of our body, so, our hair cells, our skin cells, blood cells, brain cells, the DNA code needs to be wrapped up really tightly into what we call chromosomes. And we have 23 pairs of chromosomes comprising our DNA code, so 46 in total for humans, where we've inherited one set of 23 from our mother and one set of 23 from our father. And these are identical in every single cell. So to just, I guess, give an analogy on how big our genome actually is, if we started to type out all of our DNA units, so our A, T, C, and G, if we started to type them out, we would end up with around 6,000 DNA units on an A4 page. So to then convert that into a full human genome, it would actually require 1,500 page books for our entire genetic code, our entire DNA sequence. So on average, each of our chromosome would have about 40 volumes. So our genome is really big. And just a reminder, this is in every single cell in our body. And so an important component of our DNA are our genes. And genes are effectively just little segments of genetic code, but they are critically important because although they only make up around 1.5% of our entire genetic sequence, they provide snippets of information for human traits, which is what makes us who we are. So these are our traits such as height or eye color, or even predisposition to disease. And these are all encoded within our DNA. So what genes actually do is to provide information for the making of the building blocks of our cells. So if we imagine our DNA is our blueprint. So then we would consider the next iteration of DNA, which is called RNA. It's effectively just a small photocopy of our blueprint. So providing a snippet of instructions to then be translated into a protein, which actually makes, this is actually what we make the building blocks of our cells. So we've gone from a blueprint to the actual product. A specific example to help understand is the ABO gene. So this is a gene, its name is ABO, and this is the gene that encodes the information for our blood type. So most people would know what their blood type is. So our DNA has the blueprint. It has the sequence which will define what blood type that you are. The protein that's made from our DNA code is the antigen, and this is what sticks on the outside of your red blood cell and gives you your blood type. So an important aspect of our genetic code is genetic variation. And this is actually what gives us diversity as humans. So humans, we have 3 billion DNA units, and we have around 4 million changes to our DNA. So we have humans, we have around 20 to 25,000 genes across our entire human genome, and we all have the same genes. We just maybe have little slight differences within our genes, and these are called genetic variation. And so our changes, they give rise to our diversity. So that the changes that we have are why I have brown hair and brown eyes, and why I have freckles. But if this genetic change, if this DNA change leads to a detrimental consequence, such as, I'm actually colorblind, we would consider this genetic change a mutation. And so I'm just going to give another example of types of genetic variation that we see in the human genome. So here we have a gene which leads to a protein. If we have a single change in that gene, so one DNA unit has been changed in a different person, we end up with a different protein. And this single change in the genome is called a single nucleotide variant. Or if it leads to disease, we would call it a mutation, a single nucleotide mutation. Another type of genetic variation is when we're actually missing a small chunk of our human genome or of our gene. And this is called a deletion. And what can happen here is that this gene may not actually make a protein anymore. Another type of genetic variation that we commonly study are these little repetitive sequences of our DNA. So you can see here within this gene, the section that I've highlighted, we have a TA, 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 TA repeat through our gene. That leads, this gene here makes this protein. If this repeat region is actually expanded, so it's not, we don't have 4 repeats anymore, we maybe have 10. The protein now looks slightly different. We have this larger extended little pink chunk of this protein. So these are repeat expansions, and they can be, if the repeat expansion is too large, these can be quite pathogenic or detrimental. And lastly, what actually is inheritance? So how are genes inherited? So as I mentioned before, we inherit one set of chromosomes from our mother and one set of chromosomes from our father. So we effectively have 2 copies of every gene in our body. And so we have 2 copies of every gene in our body, and then we inherit one from our mother and one from our father. So the mode of inheritance with genetics determines whether we display the trait that gene encodes for. So if it's a dominant gene, you only need to receive 1 copy from either your mother or your father to display the same trait. If it's recessive, you actually need it from both parents. So in the case in this diagram here, where we have dominant inheritance of this trait, which we are considering a disease here, the 2 offspring that inherited this red gene are the ones who also get disease. So this is just, I guess, some brief background on genetics. And so why is genetics important in motor neuron disease? And it's because when patients present at the clinic, we generally classify them as either familial or hereditary or sporadic. And it's these familial hereditary cases that demonstrate why genetics is so important in motor neuron disease. So what we see here, this is what we call a pedigree, which you could also think of as a family tree, where we have many generations of individuals, and we can see that the shapes that are black, so the black circles and the black squares, these indicate individuals who are affected with motor neuron disease. So looking at this pedigree of this family tree, we can see clear inheritance of the disease from generation to generation. And this is telling us that there is a gene mutation that is held by all of these people who have motor neuron disease in this pedigree. Now, I dislike presenting the classification of MND patients as this binary classification because it's not realistically what we see in the clinic. We tend to see what we would call a spectrum of inheritance. So we have true large hereditary families, and then we have true sporadic cases, but we also see families that we call low penetrant families, where it looks as though the disease may actually skip a generation. Or we have sporadic cases that have a mutation that we know cause the hereditary form of disease. So these are patients who have presented at the clinic with no known family history of motor neuron disease, but actually have a gene mutation that we know causes motor neuron disease. And so this is why it's important, it's a critical aspect of motor neuron disease. So understanding the genetics, it's what I've done for 16 years. So the first gene for motor neuron disease was discovered back in 1993. So that was 30 years ago. And since then, global motor neuron disease researchers, we've worked independently and we've worked together, including, our research program that we have here in Australia, to find more than 30 genes that either cause motor neuron disease or are associated with motor neuron disease. And so you can see that we've had a rapid, this is a slightly out of date figure, but we've had a very rapid increase over the last decade. These 4 genes here are what we would consider the 4 main motor neuron disease genes. So these are SOD1, TARD-BP, FUS, and C9orf72, the 4 major MND genes. And so this pie chart here really, it summarizes my whole academic career of research in Australian MND, and it summarizes the mutations that are present in our MND families here in Australia. And I think a really important aspect of this is the C9orf72 mutation is also the most prevalent cause of sporadic motor neuron disease in Australia. So briefly, I'm yet to come to why genes are so important, but how do we find genes? So we have families present at the clinic and we want to be able to find what is the genetic cause of disease in their family. So ideally, we collect DNA samples from many individuals from the family. And what we're looking for within these individuals is one genetic change in their 3 billion DNA units that all of these individuals share. So historically, we would manually look. So we'd be able to pinpoint maybe one of these 40 volumes on a chromosome that this mutation was sitting in. We would then perform DNA sequencing to see if there was a change in these individuals that had motor neuron disease compared to people in the family that did not have motor neuron disease. Now we actually use computers. So that's why our research field in genetics is now what we would call genomics as well as bioinformatics. So we sequence entire human genomes now. So we get full DNA sequence code, so 3 billion DNA units, and we write software, algorithms, programs, bioinformatic pipelines to be able to pull out the single genetic change in 3 billion DNA units that is shared amongst all these individuals that is different to people that don't have motor neuron disease. So this is how we find mutations now. So this specific example here is the most common SOD1 mutation in Australia, the one that makes a change to the protein at position 114. So importantly, we need to consider sporadic cases when we're thinking of genetics, and that's because both familial and sporadic MND cases can have gene mutations in these major major motor neuron disease genes. So here is the SOD1 gene. So this is the full genetic code for the SOD1 gene, and you can see it's made up of A's, C's, G's, and T's. And so this gene itself is only 465 DNA units. And over the last 30 years, mutations have been found all across the gene. So this is a schematic showing the full length of the gene and all of the different mutations that have been found in this gene that either cause familial motor neuron disease or sporadic motor neuron disease. So it's quite striking, and this is why SOD1 is one of our major genes that so much research has focused on. Interestingly, C9orf72, the most common genetic cause of motor neuron disease, so both hereditary and sporadic, it's only a single mutation, and it's not actually in the gene. It's just upstream of the gene. But again, this mutation is present in both familial and sporadic motor neuron disease cases. So over the last 30 years, as I mentioned, we discovered maybe around 30 disease genes. But of these disease genes, there are around 1,000 genetic variants or mutations present in the DNA sequence of these genes. So motor neuron disease researchers across the world collate this information into databases. So two really important online resources are ALSOD, which reports mutations that have been found in motor neuron disease. So these are literature-derived mutations. And then we have ClinGen. So this is recently a new initiative from the NIH. Is to obtain evidence from the literature on the strength of a disease gene, so the clinical relevance of a gene to a specific disease. And so I guess finally, why are disease genes important in MND? And for those that have a family history of MND, every new gene that we find, we're able to add to diagnostic panels. So that can give an answer in the clinic to patients when they present with motor neuron disease in 2/3 of cases. And it also offers the opportunity in specific cases for pre-implantation genetic diagnosis through IVF to stop further inheritance of motor neuron disease. Generally across motor neuron disease, by understanding disease genes, it allows us to mimic the disease in the laboratory, but also better understand the biology underlying disease. So you may remember that the blueprint, the DNA, leads to a protein. If the DNA is defective, it may lead to a defective protein. And if we understand what that protein does, it allows us to maybe find targets for therapies or just better understand what's actually happening in motor neuron disease. And really importantly, finding disease genes is important for development of gene therapy for clinical trials. And so I'm briefly going to go over what gene-based therapies actually are. So to summarize, it is just the delivery of genetic material to human cells. This is what a gene-based therapy actually is. And the ways the outcomes of gene-based therapy can be to introduce a functional copy of a dysfunctional gene. So if your gene is not working properly, you can replace it. And this is the case with spinal muscular atrophy, which is for this particular treatment, it's for a congenital form of lower motor neuron disease. And so a single injection with this virus, so this is a proper medical use virus for this gene therapy. So the adeno-associated virus, an AAV virus, encapsulates a copy of the functional gene. This is one injection is given to a patient to then effectively replace the dysfunctional gene that's not working. So this was approved, this was FDA approved 4 years ago and was really the first key step in gene therapies for motor neuron disease. A second gene-based therapy is to silence harmful gene expression. So if in the first instance, your gene's not working, in the second instance, the mutated gene and the mutated protein is actually doing something bad. So we need to try to stop it. And this is what an antisense oligonucleotide effectively does. And this is what tofersen does. So it's an antisense oligonucleotide. So this is a gene-based therapy. This is a synthetic form of DNA. It's a synthetic form of DNA that targets the defective gene, which I'll show you in the next slide. And the third type of gene-based therapy, this is where we would permanently edit the human genome. So this is using specific targeted technologies that fix your DNA mutation. We don't have clinical trials of these at the moment, but this is hopefully an exciting future avenue of gene-based therapies. Okay, so now to come to one of the most exciting outcomes of MND research in a very long time is the FDA approval of tofersen. And so this is an antisense oligonucleotide against SOD1. And SOD1, if you remember, accounts for about 15% of familial MND. So when we have a normal SOD gene, we get a normal gene product, we get a normal protein. When it's mutated, we get a misfolded product, which accumulates, which is damaging to brain cells, motor neurons in the brain. An antisense oligonucleotide is a synthetic DNA that will bind only to SOD1 in the human genome. So what it will do, it effectively blocks the gene, it blocks the mutant gene, therefore blocks the formation of this toxic product. So this is, it's a very simple concept. It's a very simple concept, but it's so exciting that this has finally gone through as a therapy. And so what genes are currently being targeted either in clinical trials are 3 of our 4 main genes, so C9orf72, SOD1 and FUS, and also another gene, ataxin-2. So that's the hereditary form of motor neuron disease. What about the sporadic form? What about genetics in sporadic MND? And the only thing I could really say is that it's complex. The genetics of sporadic MND is complex. We know that there is an underlying genetic basis to a large portion of sporadic motor neuron disease. Not all, but there is evidence of this underlying genetic basis. So we suspect that it's a combination of genetic risk as well as environmental factors that are leading to sporadic MND. And so how we find these genes with sporadic MND is is we need large cohorts. We need really, really large sample numbers, so tens of thousands of samples to effectively do statistical tests. So is a specific genetic variant more prevalent in motor neuron disease versus a non-motor neuron disease control? And we're part of a large international sequencing consortium here in Australia. We're part of Project Mine. And the aim of Project Mine is to get genome sequencing data for 15,000 motor neuron disease patients worldwide to really understand the genetics of sporadic motor neuron disease. So just quickly, two recent studies that we've conducted in my research program on sporadic motor neuron disease. The 1,000 genetic variants that I described before that have been associated in the literature with motor neuron disease, we looked at a large cohort of around 600 Australian sporadic MND patients. And we found that of these 1,000 variants that had been reported, around 30% of sporadic MND cases in Australia may have or do have one or more of these genetic risk variants. And we also found that the more genetic risk factors you had, the earlier age of onset was sporadic MND. And finally, some very recent research that's come out from my team is looking at this more complex genetic variation, which are called repeat expansions. So this is where you have a repetitive DNA sequence that at a certain length is considered normal. We all have them. But once it starts to get past a certain threshold, it becomes detrimental. And the most common genetic cause, C9orf72, is actually a short tandem repeat expansion in C9orf72. So it's not just a normal single change, it is a repeat expansion. So we looked at repeat expansions that are known to cause other motor degenerative diseases. And so we looked at about 20 different diseases. And in particular, this gene that we found, ataxin-2, we found that MND patients, a lot of them don't have a normal range, they don't have a very highly expanded range, but they fall in this middle, what we would call intermediate range. And this gives an increased, we suspect gives an increased risk of developing motor neuron disease, having this increased repeat expansion. And so ataxin-2, this was the 4th gene that's currently having gene therapies developed. So that's very important in remembering these are in sporadic motor neuron disease patients, not familial. And so, as I said, as we suspect, repeat expansions throughout the genome may increase the risk of sporadic MND. And so finally, to conclude before I hand over to Dr. Ashley Crook, that the information snippet of our DNA, which is our genes, encode really important information. And detrimental changes can lead to motor neuron disease, whether they're small changes in the genome or maybe larger expansions throughout the genome. Individuals that don't have a family history of disease are also, there is genetic risk for individuals that have sporadic motor neuron disease. But I guess something positive, a really exciting outcome that comes from genetic discoveries is that the genetic discoveries are really they're driving the development of therapeutic treatments. So that's it. I'd now like to hand over to Dr. Ashley Crook. Thank you.
Thanks, Kelly. Always great to hear from you.
Mm-hmm.
I will just set up my screen. So thanks everyone for the opportunity to speak this week in particular, MND week. So following on from what Kelly's talked about, I'll be talking a bit more about the practicalities of genetic testing and counselling for MND, and I guess some considerations for those who are thinking about testing or who would like to know a little bit more. And I am sure we'll have some time for questions towards the end. So what is genetic counselling? Well, it's a communication process. It incorporates 4 things: interpretation of family and medical histories, education about the condition, support, resources, counselling to promote informed choice, and support to encourage the best possible adjustment. So I'm a genetic counsellor, and we are trained health professionals to provide all 4 aspects. But what's important to know is that genetic counselling is provided by many different health professionals. who provide genetic counselling and testing. Sometimes my role as a genetic counsellor is providing specialist kind of counselling, for example, in predictive testing, which I'll talk about a little bit later. So as Kelly has alluded to, genetic counselling and testing is emerging in the care of all people with MND, not just those with the traditional familial family history. And it was really great to have the highlight of the spectrum of inheritance, really showing that it's not so binary as, you know, you fit into a familial, you fit into a sporadic family tree. And what the latest research has suggested is really we're missing out on identifying people who do have high-risk genes or high-risk genetic faults if we only test people with a certain age of onset or certain family history. Importantly, genetic counselling kind of goes hand in hand with genetic testing because it's an opportunity to discuss the option of testing and also DNA storage, which I can talk about again a bit later. Testing should never be mandatory and should only be available to those who wish to know their results. So there's a few different types of genetic testing. There's diagnostic testing, which is the first test. And then if a high-risk genetic fault is confirmed, so that's usually a single genetic fault that causes an increased and quite high risk of developing disease over a lifetime, then you might consider the opportunity for other family members to undergo predictive testing or reproductive testing. So just about the testing types in a bit more detail. Diagnostic testing is the initial search for a pathogenic variant or a mutation or a genetic fault or a genetic change. They're all the same kind of terminology used to describe, you know, finding a fault within a gene that causes a high lifetime risk of developing disease. So for someone who undergoes diagnostic testing, usually what happens is you would look at least at one entire gene and do a search, or look at multiple genes to detect whether there's a genetic change present. It's best on someone with MND if possible, because then there's fewer limitations of the testing. And really there's three possible results from testing. So the first and the most common result is that no genetic change is confirmed. Secondly, there's uncertain significant results where we might find something that we don't fully understand what it means. And then there's a positive result, and that's a result where we actually find that we have confirmed a genetic change in that person. So if it's negative or uncertain significance, there's no further testing available in the family, particularly if you've tested the most appropriate person. It means that perhaps our current technology may not be able to pick something up, or maybe there is no underlying gene to explain that person's personal history of disease. But certainly testing is not perfect, and as Kelly showed, we're learning more and more about the genetics of MND every day, every year. So our knowledge certainly might change in future, and it might mean something different. For those who have a positive result, it could provide an explanation for why disease occurred, or at least a part explanation. You know, maybe there's this gene plus some other factors that might cause someone to have developed disease. Sometimes those other factors are difficult to determine. As Kelly said, it might be other genetic risk factors. Maybe there's other environmental factors or chance factors that come into account, which are difficult to know and understand at this point in time. If we do identify that there is a genetic change or pathogenic variant in a person, then we can often offer predictive or reproductive testing, particularly if it's a well understood genetic variation and we have kind of a good way to interpret the meaning of the results. For a select few people, there is the possible access to clinical trials, as we've talked about tofersen, and I'll talk about a little bit further later. So just on to predictive testing. So this is the test that's available for someone to predict their likelihood of developing disease in the future. So it's a yes or no answer. Have they inherited the risk known to be present in their family? If it's yes, that person is at increased risk over their lifetime. For some particular genetic variations, that risk is close to 100%. For others, it might be less than that. there could be access to clinical trials, and as well as giving information about risk for themselves, it also clarifies risk for children or future children. And as Kelly showed, there's dominant inheritance where it's often, for most of these genes, 50-50, whether it is passed on to the next generation for each child. If someone undergoes predictive testing and the answer is no, they have not inherited the MND risk known to be present in the family, it's quite possible their risk might be reduced back down to being the same as the general population. And it also means they can't pass that risk on. As I noted before, if there's any uncertainty, predictive testing may not be available, because we want to make sure that we are providing as much information as possible for people who are deciding about predictive testing, because often people are making decisions to undergo predictive testing to consider different big life decisions like family planning, and to guide how they might live their life. You also can't unknow this information, and we know that actually the majority of people don't proceed with predictive testing at this point in time. So genetic counselling, ideally by a specialist genetic counselling service, would be ideal. The sword of Damocles is a way that some families have described it, feeling like there's a sword hanging over your head, knowing that you are at risk of developing disease in your future, but not knowing if, when, and how disease might develop. So it can be quite complicated, the decision whether or not to proceed, but certainly genetic counselling is something that is available without any obligation to proceed with going ahead with testing. And then there's reproductive testing. So I'm really summarising this very briefly. There's a few different options where you can test embryos through IVF or test a pregnancy directly. And the goal of reproductive testing would be to prevent the inherited risk being passed on to future generations. So if you're testing a pregnancy, it might be considering termination of pregnancy, for example. There's also the option of exclusion testing, which is always worth me flagging. So this means that if someone's from a family where there is an inherited motor neuron disease gene, and they're at risk of carrying it, but they don't want to go ahead with predictive testing because they can't see that there are any, the benefits outweigh the risks, then they may wish to consider exclusion testing, which is a way to conduct reproductive testing without knowing your own risk, without knowing your own predictive testing results. And similarly to predictive testing, specialist genetic counselling would be recommended for these aspects. There's lots of pros and cons and lots to work through, and we want to make sure that for anyone going through with predictive or reproductive testing, they've made the right decision for them at the time to proceed or not. So very exciting, as Kelly said, we were talking about this earlier as well, is the FDA accelerated approval for tofersen. As of April. So the accelerated approval basically means that through a biomarker, which is NFL, or plasma neurofilament light, they noted that people who took tofersen had a decrease in the NFL measurement, and that's believed to be associated with changes in nerve injury or neurodegeneration. So the idea is that if someone continued long enough on tofersen and their NFL measurements continue to decrease, then you would eventually end up with changes in clinical function or survival. The studies are ongoing, so although we don't have all the data yet, people are participating in trials to understand more about that. It is a spinal injection administered every 2 weeks and then every 4 weeks. And in terms of availability in Australia, so for people who have MND and have a SOD1 genetic fault or pathogenic variant, there is the expanded access program. So that's something that your doctor who cares for you and manages your MND could consider contacting Biogen to find out your suitability if you have MND and you have a SOD1 pathogenic variant. For people who have a family history of SOD1 and have undergone genetic testing, there is the option to participate in the ATLAS study. So this is an ongoing phase 3 trial, which involves a placebo arm as well as a treatment arm. And that's really to kind of ideally reach a point where in the future we can demonstrate a clinical benefit of having tofersen. So this is for people who do not have symptoms yet. They only have specific SOD1 variants, and one thing to flag is there's one SOD1 variant called I114T, which is one of the most common ones in Australia, and it's not included on the list of variants that is covered in the ATLAS trial. And there also needs to be neurofilament light measurements of a certain level to be able to be enrolled in the trial. And the only site currently conducting that trial is at Macquarie University. So I just wanted to mention those and would suggest if you wanted to look into it further that you consider contacting your care team about that. Kelly also talked about some other trials, and yes, I would expect that there would be more other trials available soon for other genetic variants, but not for all of the genetic variants that we can currently find with our testing. As Kelly said, there's really only a few of the genes that are at the point where there may be trials becoming available. So I have worked clinically in this area for a while, and I also just finished my PhD. So just very quickly, I will go through some of my PhD projects and kind of what it came down to. It was all about genetic counselling considerations in testing for MND and FTD, which is a related condition. So I started with some literature reviews really to scope out what we knew, and that was looking at how we provide genetic counselling for all late onset neurodegenerative diseases like MND. And then the focus became about, well, what are patients and relatives' experiences of genetic testing and counselling? From there, there were several gaps that I identified, and I wanted to conduct some interviews with people who had experienced genetic counselling and testing in Australia to understand what their experiences were, what their preferences were, and how things could be improved. And then from there, I conducted a survey with experts in the area. And when I say experts, I mean people who had experienced genetic counselling and testing for themselves or in their relatives, people from MND associations and health professionals who also provide genetic counselling and testing. So from there, I came up with, well, together with the people that I worked with on the survey, we came up with 16 items that were considered important by more than 80% of participants as part of the genetic counselling process. I'd love to talk your ear off about all of them because this is just the summary here. But I'll just focus on a few now, really, just to highlight some of the things that might come up or could be considered as part of the genetic counselling process. And it's also important to note that this might seem like a lot. Some people might be listening and say, well, I just want to have the test and then I'll just deal with the consequences. And certainly there is flexibility with this list. It doesn't mean that you have to cover all the points of discussion beforehand. It's more it should be informed by the person in front of you and the needs of that person and what they wanted to discuss, although there are some key things which are quite important. So for example, use, privacy and storage of results. This is kind of how it looks in detail, the overview. So you can see that it's quite specific that before testing is arranged, providing information and gaining consent for the use, privacy and storage of results now in the future is important. And this includes whether information could be shared if that person passes away with certain family members, and kind of how that information is used in the future. And that's really important because we know that genetic testing information is not only important for the person having testing, but potentially for other family members as well. Excuse my little tickle in my throat. Then just to highlight a few others, so implications for the person with MND and others. So one of the suggestions would be to consider and discuss and explore the possible practical, clinical and emotional implications of testing. Think about insurance, financial, life and family planning implications, whether there are any changes to management, whether there are clinical trials available, and how someone might respond emotionally to each possible genetic testing result, and the fact that genetic testing results may provide the opportunity for other people to undergo testing and how that might be. So genetic testing in many ways is a family affair. So it's something that we certainly encourage consideration of, not only the impact on the person with MND, but also other family members. And I guess on that note, the condition being investigated, it's important to know that there can be varying disease clinical symptoms. So some genes, like C9orf72, for example, in some families, you see family members with frontotemporal dementia only. So the importance of being aware of the conditions and the spectrum of diseases that could come up as associated. Also how penetrant a genetic variant is. So Kelly talked about there being like high risk genes and also like risk variants. So variants where maybe a combination of genes cause someone to be at increased susceptibility of developing MND. There's different things to think about that and different implications for that person and their family. And the genetic testing available, so not all testing is the same. So you might have a genetic test for C9orf72 only, which is the most common genetic form of MND, but it then doesn't allow the opportunity to consider whether SOD1, for example, is present. And some people might choose to only test for SOD1, for example, because there is a clinical trial potentially available. So it's worth knowing about, what exactly is being tested and the pros and cons of more and less information. Finally, the approaches to practice. So making sure there's information in several formats and supporter information resources. Just about that, I thought I'd finish with some useful resources that you might want to screenshot. So I'll just detail them all here. So first is the Les Turner, My ALS Decision Tool. That's from the USA. That is a decision tool which could be useful for people considering diagnostic testing. It might walk you through some of the pros and cons of testing and considerations. Then you've got the Health Talk website. This is a website in the UK where interviews were conducted with people affected or, with a family history of inherited motor neurone disease, talking about, their experience of finding out about the genetic disorder, how they made decisions about testing, and what factors they thought about. So can really be useful sometimes to hear about other people's perspectives. There's also MND decision tools. That's an Australian website. And one of the decision tools is about predictive testing. And the other thought that I had that might be interesting is this Connecting ALS podcast. So there's the Tofersen and the Future of ALS Drug Development episode, which really gives a bit more of a detailed overview about Tofersen and ASO therapies, and might be a useful thing if you are interested in finding out more. Thank you very much. So I've got my email here and Twitter. Happy to hear from anyone. Hopefully we've got some time for questions now. Thanks all.
Great. Thank you, Ashley and Kelly. That was a brilliant overview of obviously what's becoming a more and more of a critical area in MND. We've got quite a few questions here, so I'll jump straight into them. So a question here about someone has other members of a family who've all passed away from MND. Can they have genetic testing? And perhaps a wider question is also, does that vary from state to state, or is there uniformity across Australia?
Yes, it would be great if there was uniformity. So for these kinds of discussions, often they are had in a public service genetics clinic. There are genetics clinics in all states, so there would be access to them. In saying that, there is different availability and funding for genetic testing and perhaps different attitudes to the benefit of testing. For someone who has a family history of MND and, ideally in the perfect situation, you would be wanting to test someone who had MND themselves, because we know that that's the most accurate way to do testing. But we know that MND is unfortunately so awful that sometimes there isn't time to do that, or it happened so long ago that it's not available. So for someone who has a family history, that includes probably multiple relatives with MND, it's definitely an option to consider testing for themselves. They might require a referral to a local genetic service to have that discussion. The other thing is that there are sometimes genetic research studies that someone may have participated in when they were alive, and that could also be another opportunity to see if there is any DNA available, although there's certainly never a guarantee of that, and there's lots to consider in that regard. So it'd be a matter of finding a health professional who could walk you through that. And there is a website called genetics.edu.au that has all of the genetic services available in Australia that might be of use.
And with all these testing centres, are there usually genetic counsellors kind of associated with them or attached, you could say?
Yeah, great question. So most public genetic clinics, which is the majority of genetics clinics in Australia, include geneticists, doctors, as well as genetic counsellors. And I know, in my experience working here at Austin Health, I probably have most of the discussions with people who are considering that kind of a testing. It's definitely valuable to have that. And even a geneticist, even though they're not a trained genetic counsellor, they certainly have a lot of crossover with genetic counselling.
Thank you. I have a question here about what proportion of people who have the C9orf72 mutation will actually go on to develop MND. Do we know the penetrance?
Yes, I'm going to pull up a picture that I'm going to share my screen. So this is from a research study that I conducted back in 2013 when we looked at all C9orf72 cases in Australia. So I'm just checking. You can see my screen? You can see this picture?
You've got your desktop at the moment.
Oh, sorry. Yep. Okay.
Yep.
Yep. So this is from a study that we conducted back in 2013. So this shows age-dependent penetrance across ages of. So these are specifically patients that have a C9orf72 expansion. So this is including pre-symptomatic individuals. So what this graph means is that, say, at the age of 70 years, if we go up to 70 years and across, so for females at the age of 70, if you have a C9orf72 repeat expansion, there is a 70% chance that you will have developed MND at that time. So if you then extend that, say to age 60, that drops down to 40%. So at age 60, only 40% of mutation carriers will have developed MND. And then by the time you get up to, say, 80, it's up to just over 90%. So for the samples that we had, it never reached 100% for females. It did reach 100% for males, and that was by the age of 85.
Okay, so like everything, it's never simple. It changes constantly. Right. And just leading on from that, so there's a patient here who says a pre-symptomatic carrier, they're involved in the DINAD study, which isn't underway anymore. Are there any other genetic studies where people can enroll and contribute to trying to boost our knowledge of genetics?
I know we would be at Macquarie University, we collect pre-symptomatic participants into our biobank all the time, including longitudinal samples. So please feel free to send me an email after this, because we would be very interested in being able to collect samples. So thank you for being open to donating samples.
And can people do that if they don't live in New South Wales or can't get to Macquarie? Can they do it remotely?
Yes, we do arrange remote collections, so we are able to do that. Yeah.
Okay. So another question around the testing here. So do you have an idea why the majority of people do not decide to proceed with all the way with genetic testing? What aspects put them off? Is it financial life insurance implications or something else?
Yeah, I think often it comes down to the predictive testing. So for this, I guess only up until recently, there's the potential of a clinical trial available. So a way to kind of potentially do something from a medical perspective, act on the information that comes from knowing that you are at high genetic risk. We know that there can be negative counselling, psychological implications of finding out one way or another, increases in, depression and anxiety, particularly in the short term for people who make the decision to proceed with testing. Insurance and, financial implications can play a role. So certainly in Australia, life insurance, there can be implications of genetic testing for life insurance, particularly for someone who is unaffected, who is finding out whether they have an increased risk. There is a moratorium, basically meaning that you can be covered for a minimum amount, for example, $500,000 of life insurance. There's different amounts for different types of insurances. If you go ahead with testing and you're asymptomatic, for example, and you already have an insurance policy in place, then you it's renewable. So I guess that's something that people might consider before they go ahead with testing, is looking into their insurance and putting life insurance possibilities in place if they know they are at risk, but don't necessarily have the know that they have the genetic form of the genetic risk for developing MND.
And are you obliged, I might have missed this, are you obliged to inform your insurance if you have had a test and you know the results?
If you have a current policy that you have had before you applied, it's guaranteed renewable. So you're not obliged to declare that. The risk would be if you applied for insurance after you had testing and you knew that you had a genetic risk, but you didn't disclose that at the time, and then you wanted to do a claim related to MND, then there would be probably a decent likelihood that you would not be covered because you were not honest at the time of applying. So it's all about when you apply or when you upgrade your policy. If you have a current policy in place, it's guaranteed renewable. In saying that, I guess for some people who go ahead with predictive testing, they might have at the time of, applying for insurance said that they had a family history of MND, and then maybe be charged higher premiums or not covered for certain things if they got a large policy. If they test negative, it may be beneficial for them to actually go back to their insurer and say, hey, there is this MND in my family, but I'm not at risk. So pros and cons, but really, I guess the main message is it's guaranteed renewable, and we would be, as part of the genetic counselling, it would be suggesting that people look into their insurances before going ahead with having a blood test and doing the predictive test.
Thank you, Ashley. And this is probably a question for both of you. If someone has MND but they don't have a family history, is it still worth considering testing? And if so, are they eligible for testing, or what's the best way to go about it?
Yes. In short, I think, there's a lot to consider, and I know, through Macquarie, for example, genetic testing is kind of becoming a bit more commonplace, that it's offered to anyone who comes through the door diagnosed with MND kind of thing. That is different in different clinics. So I also work at the Austin, a public clinic, and we have a certain criteria that people need to meet. So they need to be under a certain age of diagnosis, or there needs to be a family history to cover the test. In saying that, testing used to be thousands of dollars, and now testing is something that is a bit more accessible, maybe between $550 and $750. So some people still elect to pay the out-of-pocket costs if they are not covered.
So if you're willing to pay for it, there's no restrictions. You can get tested regardless of your status.
Yeah, you would probably, ideally it'd be an initial chat with the neurologist who is managing your MND. because there can be wait times with genetics. So if you can access testing through the neurologist rather than coming to a genetic counsellor, then that could be useful. But in saying that, it can be really beneficial to have a genetic counselling conversation, waiting for that opportunity too.
And for those of you asking about other studies, there's a study called the TONIC study at the Parent Institute in Perth as well, if you're based over in WA. They're also collecting samples and can arrange for blood samples to be taken. And just on that, to be genetic tested, is it usually just through a blood sample or?
Yeah, or saliva. Yeah.
So it's very non-invasive as well. This goes back, probably a question for Kelly here, or well, I'm sure you can answer as well, actually. But what's the child's risk of developing MND if one parent has MND, but where no other family members have had MND, such as grandparents, aunts, etc.?
So it's a little bit complicated. Nothing in MND genetics is ever simple. If a person has presented at the clinic as a sporadic, so there's no evident family history, but they do have a known mutation such as C9orf72 or SOD1, then it would be a 50% chance of passing on that gene mutation to offspring. Again, depending on penetrance is whether the person would develop MND or not. But if the person would be considered a true sporadic, so there's no genetic risk that has been identified, and this is off the top of my head, I believe the overall lifetime risk of MND is 1 in 400, and I think it increases to 1 in 200 or 1 in 300 for family members of sporadic MND. So that's lifetime risk. It's lifetime risk.
Thank you, Kelly. Another question we have, did you have a similar graph that you put up for C9orf about the penetrance of SOD1 and also for other genes? Is that information out there?
Yes, we published that in 2016. So because I'm not connected to the university server, I don't have access to the full paper. But yes, so Dr. Emily McCann, my PhD student, she published a paper called The Genetic Landscape of MND back in 2016, and that gives the age-dependent penetrance across C9orf72, SOD1, I think 5 different variants, and SOD1, TARDBP, and FUS. So yes, the information is out there. Yeah.
Are they generally, sorry, just a quick question, it's my fault for introducing the term penetrance. Can you just quickly explain penetrance in non-technical terms?
Ashley, would you have a good definition? It's really...
It's kind of related to the likelihood of developing disease. So we talk about high penetrance genes. So those are genes where there's a high likelihood of developing disease. And then there's lower penetrance or variable penetrance genes where maybe it's uncertain or there's a lower likelihood. That's what I'd be thinking about.
Yeah. So, sorry.
No, I was just going to say it can depend on age as well. So similar, I guess like the graph that I showed, penetrance at a very, even though it may be a highly penetrant gene like a SOD1 mutation, at the age of 20, it's unlikely that a person would be affected. There would be a very, very low percentage that a person would be affected with MND at the age of 20, all the way through up till 90.
And we can put some links up on our Facebook page tomorrow for people who are interested in those other studies about the other genes for people as well. But is it right then that probably the FUS SOD1 mutations, they're not as penetrant, or you're less likely to develop MND if you carry those genes, or is that not the case?
No,
okay.
not in my experience of what we've looked at, so no.
Okay.
Seems to very much depend even on the specific variation or spelling mistake within that gene as well.
Okay. And just a last question. This is one from me actually. So I'm just wondering if I think you were mentioning, Kelly, that you're finding more and more that patients who were previously thought to be sporadic are carrying genetic mutations. And so we're still using the term 10% familial genetic and 90% sporadic. I mean, I've seen data, I think, from Ionis saying that the shift is more like 15 to 20% of people with probably causative mutations. Would you agree with that sort of, we should be shifting that's not the language, certainly the numbers we're using? You think 10% is really accurate still?
Oh, well, I guess normally when we refer to 10%, we're talking about like clear family history of disease. So even though we're seeing genetic, risk factors or SOD1 and C9orf72 mutations in apparently sporadic cases, they still aren't presenting with a full family history of disease. So I think for classification purposes, it's reasonable, because again, if a person presents at the clinic and no one else in their family has it, there's no real reason to suspect they might have, hereditary motor neuron disease. And so I know there is talk amongst like neurologists and geneticists worldwide that they're not liking this binary classification, but
yep.
it is useful. But no one's come up with a good solution yet for what to do. So
yep, watch this space,
yeah.
as with everything.
Mm-hmm.
Well, thank you both. That was a fantastic session, and thank you to the audience as well for diving in with lots of really great questions. And I think, as Ashley said, it's MND Awareness Week, and tomorrow is Global MND Day. It's brilliant for us to be able to showcase the world-class research we have in Australia. We're very fortunate to have the whole research community here in Australia. So thank you both, and very much appreciate you coming on tonight. Just a bit of housekeeping for those watching tonight. Everyone will receive a link to the recording, everyone who registered, so please feel free to distribute that across your networks or watch it again. Be well worth watching again, so everyone can appreciate the fantastic research and great information coming. This will be up on the MND Australia website as well, with this and all our previous editions, and in from last year and the year before. Just look under the news and updates section. And we've also got our regular monthly research updates, research directions that come out now as well, to catch up in some other more recent advances. So that's all for tonight. Thank you again. Thank you both, and everyone else. Please enjoy the rest of this very cold evening, and we'll see you all again in a few more weeks for the next State of Play. And good night all.
Thanks. Bye, everyone.